Hi, this is Professor Van Landingham. Welcome to RTE 2385 Radiobiology. This micro lecture is intended to give you an overview of what to expect in this course, also to go over some concepts from prior courses that I'd like you to review prior to beginning this course. Let's start with the definition of radiobiology. Radiobiology is the study of effects of ionizing radiation on biological tissue. What we do know is any type of ionizing event is going to happen at the atomic level of the structure. Atomic interactions take form of ionization and excitation of the orbital electrons, and it results in deposition of energy within our tissue. Energy deposited in the tissue can cause molecular change, which ultimately could be measurable. An abnormal molecule can cause impairment or dysfunction, or could result in cell death. This process is reversible. Our body is pretty amazing, so most ionizing events can be reversible by enzymatic repair, but there is a possibility that the cell could die from an ionizing event. This next slide I want to emphasize is a theory or a hypothesis. Radiation hormesis or radiation homeostasis suggests that low levels of radiation, less than 50 milligray or 5 rads, provides a protective effect by stimulating molecular repair mechanisms. All living organisms evolve and exist in a sea of ionizing radiation. There's internal radiation, there's external, there's background. And so the general belief is low levels or low doses of ionizing radiation over a long period of time will result or produce detrimental effects. And we're going to talk about some of those detrimental effects that could be caused by low levels of radiation throughout this course. But within the last couple of decades, some pioneering scientists have reported that low levels of ionizing radiation actually have proven to be harmless. Not only harmless, but they actually have provided a beneficial or a hormetic effect. So I think this is very interesting. X-rays were discovered November 8th, 1895. So in the big scheme of things, this hasn't been that long that we've had to study this. So I think in the future, we're going to hear a lot more about radiation hormesis or radiation homeostasis. Okay, so let's continue. In order to understand about radiobiology, we have to understand the 3 categories of human response to ionizing radiation. So here are the 3 categories. We have early, late, and embryo fetal. And I'm going to go through each of these. So the first one I want to go through is early acute, and I want you to associate the term deterministic with early acute. Deterministic is the radiation response is going to increase in severity with increasing radiation dose. So early acute is going to happen typically within minutes or days after exposure to radiation. This is typically associated with accidental exposure, and this is very difficult to study. So any type of deterministic early acute comes from typically nuclear warfare, some things as in Chernobyl, that we're able to study early acute effects. ARS stands for acute radiation syndrome. This happens to the whole body. There's 4 characteristic stages of acute radiation syndrome, and I have a whole section or a whole lecture on acute radiation syndrome. These 4 characteristic phases are prodromal, prodromal, latent, manifest, and then the last stage is either going to be recovery or death. This happens to the whole body. We also could have some local early effects. For example, if individual's forearm is irradiated with a lot of radiation all at one time, a local effect could be erythema. I know that's a review for you. We've talked about this term before. Erythema is reddening of the skin, so a sunburn-like effect. Other local effects could be epilation, loss of hair, or desquamination, which is a sloughing off of the skin. Deterministic effects are threshold, so there's a certain amount of radiation that has to occur in order for these effects to take place. So there is a certain amount, so there is a threshold. When we're looking at early acute effects, we're looking at the formation of cells, which is cytogenic, and more specifically, the formation of blood cells, which is hematological. Late or chronic or delayed, I want you to associate this with stochastic effects. Stochastic responses increase in incidence with increasing radiation dose. So it doesn't increase in severity, it increases with incidence. So the more radiation exposure you have, the probability of you getting a late chronic delayed response increases. This is how we base our radiation or occupational dose limits as ones that you had to memorize in your fundamentals course. It's based on stochastic effects. These are random. There is not a threshold, so you could get these effects with a little radiation or with a lot of radiation. But we know with a lot of radiation, it does increase the incidence. So cancer is a good example of a stochastic effect. Again, it's random, so you could get cancer. Are you getting it from the radiation, or are you getting it because it's genetic, or from some other, cause? Local examples of local stochastic effects would be cataracts. Cataracts is something that can occur. So the more you're in radiation, the chances of you getting cataracts, it does increase. But again, it is random. So even though you could work in radiation your entire life, you might not get them. Or, you might not work in radiation at all and had one chest X and you get them. Again, cataracts can be caused by other things beside radiation though. So again, random. Lifespan shortening, one study that was done with radiologists is that radiologists have a shorter lifespan than the general public. Could this be because they work in radiation? It absolutely could be, but it could be because they are physicians and they work in a stressful environment. Also, low levels of ionizing radiation over a long period of time, if there is a chance that it could hit our target molecule, which is our DNA molecule. If the DNA molecule randomly is hit, then what can happen is it could actually change our genetic code, which we pass down to our offspring. Again, November 8th, 1895, we haven't been studying this for that long, so I think there is going to be more information to come on genetic effects caused by radiation. Embryo fetal, I know in the clinic it's very important for you to ask your patient if there's any chance of pregnancy if they're of childbearing age. And so embryo fetal, we've talked about the law of Triboneau and Borgoni, and we know that the more proliferation of metabolic rate, the higher that is, the more radiosensitive it is, and also the younger the tissue. So obviously embryo fetal is very young tissue. So utero exposure to ionizing radiation can be teratogenic, carcinogenic, or mutagenic. The effects are directly related to the level of exposure, but more importantly, to the stage of fetal development. And throughout this course, we're going to talk about the effects on each stage of fetal development, but I do want to mention the most susceptible phase of fetal development is during orgogenesis, and that is the 2nd to 7th week after conception. So typically, this is a time when the female patient does not know that they're pregnant. So again, it's super important for you to ask all patients of childbearing age if there's a possibility of pregnant, and if there is a possibility, then we must definitely need to get them a pregnancy test, and always, always, always shield. So again, embryo fetal is something that we're going to go into detail about at different, especially the stages of fetal development and the abnormalities that can occur. Okay, so this slide is pretty important. It talks about dose effects and the amount of a dose that's needed for an effect to occur. So in order to see a drop in cell count, meaning we're not having, we have so much radiation that our cells are, the actual count is going down. So the amount that's needed for a drop in cell count is 250 millisieverts. In order to see erythema or nausea, vomiting, diarrhea, and this is something that would occur in the acute radiation syndrome that we had talked about, or even the local of the deterministic, is 1500 millisieverts. LD 5030, I know this is review for you, but LD 5030 is a lethal dose that would kill 50% of the population in 30 days, and that's 3000 millisieverts. So these numbers, I want to compare them to some of the common x-rays or procedures that we do. So the most common one we do is a chest x-ray. So in comparison to a drop in cell count, one chest x-ray would provide 0.1 millisievert to our patient. So that is a very, very small fraction of the the lowest dose effect, which would be the drop in cell count. We're talking about something that provides the most radiation, which would be the CT of the heart. That's about 16 millisieverts to our patient. So again, not anywhere near the minimum level to see a drop in cell count. So I think that puts things in perspective. I don't want anyone to be concerned about working in the field. Okay. One of the things I need for you to review to prepare for this course is I need you to be able to describe the 7 levels of organization. I know we went over this in the RTE 1503 course, so please go back and review this. The first level, or level 1, is the chemical or the atomic. This is interactions between elements to combine into molecules, specifically the macromolecules. So there's 5 macromolecules. They include RNA, DNA, protein, lipids, and carbohydrates. So our DNA is our target macromolecule. It is the most radiosensitive of the macromolecules. So we're going to study in detail how, when the macromolecules are irradiated, how that can cause effects, specifically the DNA, and how it affects genetic information. Water is not a macromolecule, but we are going to talk a lot about radiolysis of water. Water is the most abundant molecule, and it'll play an important part or role in delivering energy to the target molecule. Because water is the most abundant, what can happen is we can have an indirect hit to our target molecule, which is our DNA. Again, it's a probability, and we're going to talk a lot about radiolysis of water as well as irradiation of macromolecules. The second level is the organelle. So these are the organs within the cell with specific functions. Please review these, know the functions of them. That's going to help you understand when we talk about irradiation of different body parts. So then beyond the organelle, we have the cell. So level 3 is a cell. It's a basic living subunit. And so each cell has a nucleus, and the nucleus is that target molecule, which is the DNA, which is the most radiosensitive macromolecule. We're going to go into detail about the DNA as well as the RNA, but how, if there is any type of impairment to that based on a hit by a free radical caused after ionization, how that can occur and how that can ultimately affect our genetic code. So we have a group of cells similar in form and function, and that's my tissue. So again, the law of Bergoni and Triboneau states that the younger the tissue, or the higher the metabolic and proliferation rate, the more sensitive it is. So I want you to be familiar with all the different types of tissue, and we're going to talk about the proliferation of the tissue as well as how it affects organs and organ system. So then we have a group of 2 or more tissues with a common function, and that is an organ. And then we have organs that work together as an organ system. So be familiar with the 11 organ systems. And again, we're looking at proliferation and metabolic rate. So if I'm talking about something that's very radiosensitive, or organ system that's very radiosensitive, actually my GI system is my most radiosensitive because it has a high proliferation rate. It's a 50% regeneration. Integumentary would be another system that's highly sensitive, not as sensitive as the GI system. We have about a 2% renewal rate in our integumentary system. The most radioresistant is actually our nervous system. So our brain is very resistant. So the metabolic rate is very slow in my nervous system. Okay, so altogether, the organ systems make up an organism, and it's a living thing considered a whole. So those are the 7 levels. So please review those as we move on to the next section, which is going to be irradiation of macromolecules. And also covered in that section, I'm going to talk a lot about radiolysis of water. I need for you to be able to follow what I'm talking about in those 7 levels of the structure. All right, I'm looking forward to continuing on with this subject, and I look forward to seeing you next time. Thank you. Have a great day.